Uh, Dear students, now we will discussing the initiation of translation. In previous video, I have discussed the different components of translation which were used as tRNA, ribosomal RNA and mRNA. Now, how the initiation process is initiating in transcription, first we will discuss in the prokaryotes. So before going in detail, we have some important clues. So we have to learn that. So first of all is the tRNA which we are using here should be charged. So how it will charged? In the presence of ATP, it will completely charged. How? Why we are saying it is charged? Because in the presence of ATP, so phosphate group is there, so it will phosphorylate the tRNA, so it is charged now. So this is the charged tRNA which we are calling it as a charged tRNA. Also act as a adapter to incorporate the further amino acid in polypeptide chains. So now we will discuss how this charged tRNA compile in the assembly for further processing. So before going in detail, we will see there are different initiation factors which were involved in the processing of prokaryotic translation. So number one is initiation factor 1, initiation factor 2, initiation factor 3, GTP, 30S and 50S, which are the ribosomal units. So in first step, formation of IF2 and tRNA which is formulated methionine complex will be formed. How this complex will be formed? Because for the initiation of the protein synthesis, we need tRNA, we need one of the amino acid and further accessories which are in the form of initiation factor 2. So this is the methionine. So methionine is the initiation codon in case of translation in processing of prokaryotic system, but this methionine having the terminal end with the amino group. That amino group were involved in the degradation of further proteins. So we need not this amino group at the methionine. So we can do what we will do. So we can do one thing. So we will create the methionine which is formylated in nature. If we formylated the methionine, the methyl group is block. Okay. Now the charged tRNA with methionine, which one methionine? Methionine which is formylated. So it is also written as formylated tRNA F lower script and methionine upper superscript. Okay. So in the presence of IF2, it will create a complex IF2 plus tRNA with methionine which is a formylated. On the other hand, this is one assembly. We also need mRNA. mRNA also come with the help of some initiation factor. Here we will using IF3 30S plus mRNA. So mRNA having the AUG. AUG is the methionine, and methionine we are using in the methionine which is adding on the tRNA is formylated in nature. So in case of prokaryotes, methionine which is act as an initiation codon, but it is formylated in nature. Okay. Next step. So first step is the formation of IF2 tRNA which is formylated complex, and here is also mRNA plus 30S plus IF3 second complex. When they two complex will merge together, they will create a next step in the form of in the presence of IF1 initiation factor and GTP. Why GTP? Because GTP is an energy rich source and it will provide energy to the reaction. So in this reaction, when both the complex will merge together, so IF3 30S from here and GTP will add AUG from mRNA sequence, as mRNA contain AUG. So mRNA sequence contain AUG, complementary of AUG in the tRNA which is UAC. They get combined together and form a complex number 2. So complex 2 is the combination of both the complexes which are formed in step 1. This is step 1 complexes which are combined together to create the step 2 next complex. Okay. In case of step 3, so here we have only 30th subunit of the ribosomal unit. So in case of prokaryotes, we have two subunit. One is 30th, another is 50th. So 30th is small subunit. So here we have 30th, but 50th is pending. So how will come 50th again? 50th will come in the situation when IF3 will released GTP will break down to create the energy, and IF2 and IF1 is also released from the system. So when 50s will come? 50s will come only in one situation, when IF3, 2 and 1 will release the assembly, which is stabilized in step 2. Okay, so once the IF3, IF2 and IF1, all the initiation factors will release the assembly, GTP will break down into the GTP and PI, the step 3 complex will be formed. So in case of complex 3, or you can say that step 3, we have 30s plus 50s, so it is combinedly 70s. So 70s is the ribosomal unit in case of prokaryotes, and it is also having 3 important sites. What they are? A site, it is known as amino acyl site, P site, peptide diol site, and E is the exit site. So P is the only site. P is the only site where the first tRNA with methionine will enter directly. So we can say that methionine tRNA which is formylated can enter direct in the P site. Okay. So methionine tRNA which is formylated in nature only can enter in the P site. Otherwise, all the amino acids which were coming one by one to form the polypeptide chain, they will come from the A site. But only the methionine which is formylated in nature will come directly in the P site. Clear? So this is the how initiation occurs in case of prokaryotes. So there are some other important factors, other important information which I want to share with you, that Shine-Dalgerno sequences, SDS is the Shine-Dalgerno sequences. Shine-Dalgerno sequences is one of the important sequences which were present in 16S ribosomal RNA. So it was reported, or it was observed by the scientists, that Shine-Dalgerno sequences was present just 7 base pair upstream from the AUG in case of prokaryotes, and they are playing very much important role to initiate the translation. When scientists remove this sequence, which is Shine-Dalgerno sequences, or modified or mutated, there is minimum chances of formation of proteins will be occurred, or if protein is formed, so that is not functional in nature. So these Shine-Dalgerno sequences which are present in the prokaryotic system in 16S ribosomal RNA are playing very much important role in the initiation of translation. Okay, so this is the initiation, how initiation occurred in case of prokaryotes system. So now we will discuss the what are the different parameters. So in case of eukaryotes and prokaryotes, very much similarities is going on in the processing of translation, but some of the parameters which are differentiated in the prokaryotes and eukaryotes, which I will discuss with you. 1 second. So should I rub it? So one of the important factor which is involved in the initiation of eukaryotic system is the tRNA. The tRNA, which is formilated in case of prokaryotic, it is non-formilated. It is non-formilated. Okay, so what is the meaning of this? So methionine, which is initiation codon, methionine which is a initiation codon, in case of eukaryotes it is non-formilated in nature, whereas in case of prokaryote it is recommended that it should be formilated. Number 2 is there are child sequences were present in the prokaryotic system, but in case of eukaryotic system, in place of child sequences, some Kozak sequences were present. Some Kozak sequences are present in case of eukaryotic system. So they are just upstream to the. Similarly, these sequences are just present upstream to the AUG. Somewhere these Kozak sequences were present. So after this AUG is there. So what is the next step is, in case of eukaryotic, so 40S plus 60S in place of 30S plus 50S. So it will combine 70S. So 40S plus 60S will create 80S. Okay, this is another difference. So ribosomal unit, which are in case of prokaryote is 70S, and in case of 80S, which is in case of eukaryotic system. So some Kozak sequences which are present upstream to the AUG, and these Kozak sequences were initially recognized by the 40S small subunit, and some of the cap binding proteins, some of the proteins which are known as cap binding proteins. C. In case of prokaryotes, RNA is simply initiated with the AUG and terminated with the TGA. But in case of eukaryote, a specialized structure is present at 5' and 3'. So 3' having poly A tail and 5' having cap sequences. And after capping, there is initiation codon, which is known as AUG. So first of all, we are saying the tRNA or the methionine, which is using in initiation of the translation, is non-formilated. Second one is there are Kozak sequences which are present just upstream to the AUG, and one of the important factor is cap. Number 3 is cap. So in case of eukaryote, 5' is also always having cap sequences. Cap sequences is 7-methylguanosine. 7-methylguanosine. So these 7-methylguanosine is recognized with the help of cap binding protein. Here we have initiation factor 1, 2, 3 and GTP for energy purpose. Here we have some cap binding protein, as eukaryotic system is very much complex, so there are many proteins. So we are not naming of that 1, 2, 3 and 4. So just we are saying cap binding proteins. So we are merging the 1, 2, 3 initiation factors. Many factors are involved in the eukaryotic protein synthesis. So simply we are saying cap binding protein. Cap binding protein will bind to the cap sequences. And they just scan this sequence. Which sequences? mRNA. This is mRNA. They just scan the sequences from 5' to 3' for the searching of AUG. And where the AUG is there, the 40S subunit will, AUG is here, and 40 subunit, 40S subunit will just overcome the AUG positions. Okay, so once again I'll saying, in case of eukaryotic system, methionine which we are using, or which eukaryotic system is using, is non-formulated in nature. The child sequences is present in case of prokaryote. They are simply replaced by Cossack sequences in case of eukaryotes. Number 3 is some cap binding proteins were involved in case of eukaryotic system. In case of eukaryotic system, as a initiation factor, they just recognize the cap sequences which is present at 5 prime region. After cap binding protein, the whole sequence will scan the mRNA sequence in the 5 prime to 3 prime direction. Once they recognize AUG sequence is present over here, they just overcome the site of AUG, and 40S subunit will sit down on the AUG. Once the 40S subunit is bind over here, the next step is addition of 6 Ds, and it will create total 80S, which is the ribosomal assembly, which is the ribosomal RNA in case of eukaryotic system. So this is the initiation of different system in prokaryotic and eukaryotic. So students, you just read out this, focus in the notes which I already provided to you, and for elongation and termination we will just wait for next video. Thank you.